Okay, we are back and we are talking about B and T cells. We're talking about how antigen is presented, uh, how we have first and second signals, co-stimulators, etc. Now we're going to talk about the activation of Cd8 T cells. Remember that with Cd8 T cells, we can have the Cd8 and Cd4 cells recognize antigen on an APC that has been ingested into an infected cell. And the Cd8 T cells recognize the antigen on the infected APC, which then results into clonal expansion and differentiation. So what is happening here is that the CD4 helper cell is assisting with its cytokines and helping the naive CD8 cell proliferate, and clonal expansion happens. There's differentiation resulting in the killing of that infected cell. So let's think about two examples. Example A, we have a phagocytes with ingested microbes. In this case, the microbial antigens are inside the vesicles. So what happens here is the CD4 effector T cells, in this case a Th1 cell, would connect with a macrophage. There would be cytokine secretion, and we'd have two things happening. We'd have macrophage activation and the killing of the ingested microbes, and then we'd have inflammation. Think of another case here where we have an infected cell with microbes in the cytoplasm. Here the Cd8 T-cells, or the Ctls, would then kill the infected cell. Now remember that earlier we talked about the different types of, well, the two types of MHC molecules. So the MHC is MHC1 is associated with Cd8 molecules. So let's talk about cytotoxic T lymphocytes. The Ctls are effector T cells that recognize and kill target cells expressing the foreign peptide antigens in association with the MHC molecules. In this case, it is the MHC1 molecule. So this would be important, for example, with viruses or proteins or phagocytosed microbes. Remember that most Ctls express the Cd8 molecule and are specific for foreign peptides that are displayed by self-class 1 MHC. And now we'll just give a summary of some important points regarding the mechanisms of Ctl-mediated cytolysis. First, cells killing by Ctls is antigen-specific and contact-dependent. Next, Ctls kill targets that express the same class 1 associated antigen that triggered the proliferation and differentiation of the Ctls. And third, the specificity of Ctl effector function ensures that normal cells are not lysed by Ctls reacting against the microbes. So let's add a little more detail here from when the antigen is processed to the infected cell being lysed. So the players here, of course, we have the processed antigen and the MHC class 1 being shown on the virus-infected cell. For example, in a virus-infected cell, this would be shown on the outside MHC class 1. Then you would have the virus-infected cell, which would then show the processed antigen presented with MHC class 1. And this would connect with a cytotoxic T lymphocyte with its T cell receptors, and then we would have the infected target cell lysed. So what happens in the first section is that a normal cell would not trigger a response by a cytotoxic T lymphocyte, but a virus-infected cell or a cancer cell would produce abnormal endogenous antigens. Next, the abnormal antigen is presented on the cell surface in association with MHC class 1 molecules. Cd8 T cells with receptors for the antigen are transformed into Ctls. And lastly, the Ctl induces the destruction of the virus-infected cell by apoptosis. So again, just to repeat what has been said before regarding the differentiation into effector cells, the differentiation of mature T cells is the process that converts naive T cells into effector cells that perform various functions. And this process depends both on antigen-induced activation of the T cell and on other stimuli, principally cytokines produced at the site of antigen recognition, and that for CD4 T cells, they differentiate into two types of cells, Th1 and Th2 effector cells. There's also Th17, we'll talk about that later, and Cd8 cells, they differentiate into functional Ctls with the ability to kill target cells that express the peptide MHC these T cells recognize. Now, a moment ago we were talking about the differentiation into effector cells. Now remember that upon activation, CD4 T cells may differentiate into different subsets of effector cells that produce distinct sets of cytokines that induce different functions. So for the different T cell subsets, they are defined in part by their capacity to produce different sets of cytokines. And the first subsets to be defined are referred to as Th1 and Th2, so of the CD4 type cells. Now the principal cytokines produced by Th1 CD4 cells are TNF alpha and IFN gamma, and the principal cytokines produced by Th2 CD4 cells are interleukin 4 and interleukin 5. So just to test you, what are the principal cytokines produced for Th2 CD4 cells? They are IL4 and IL5. How about for the Th1 CD4 cells? They are TNF alpha and IFN gamma.